मैं राजवीर सिंह शेoran, एसोसिएट प्रोफेसर इन केमिस्ट्री, SRR मोरारका गवर्नमेंट पीजी कॉलेज. फर्स्ट ऑफ ऑल गुड मॉर्निंग टू ऑल द स्टूडेंट्स ऑफ MSc फाइनल. पिछले वीडियो लेक्चर में हमने एंजाइम्स की एप्लीकेशंस हैं वो डिस्कस की थी. टुडे वी विल स्टार्ट वी विल कंटिन्यू द सेम यूनिट and the same topic, enzymes as reagent in clinical chemistry. So means how enzymes roles in clinical chemistry. Today we will discuss this thing. Blood lactate and pyruvate are generally determined by LDH catalyzed method. Blood urea is occasionally analyzed using method involving urease, deglucose in blood and other physiological fluids is determined by glucose oxidase methods. These methods are specific for beta D glucose if solutions are not freshly prepared. The reaction catalyzed by glucose oxidase is utilized in test strip and clinic sticks. For screening of urine specimens, glucose oxidase and color producing reagents are impregnated into a plastic strip. The intensity of blue color obtained when dipped into a specimen gives as indication of D-glucose. This test is used in diagnosis of diabetes mellitus. D-glucose can also be analyzed by the use of glucose dehydrogenase or hexokinase. Blood cholesterol may be determined by reactions catalyzed by cholesterol oxidase. Triglycerides and urate may also be analyzed by enzymatic method. Luciferase is an magnesium 2 plus activated enzymes and act upon luciferin according to the following reaction. Luciferin plus oxygen plus ATP give rise to oxyluciferin plus ADP plus PI. So this shows chemolutions. Therefore, the reaction can be monitored spectrofluorimetrically. Luciferase catalyzed reactions can be used in the analysis of ATP as well as to determine magnesium 2 plus ion concentrations. Now we discuss enzymes and recombinant DNA technology. Means how genetic information is stored and transmitted is still unraveling. Recent advances in recombinant DNA technology allows DNA molecule to be cleaved selectively then combines with fragment of DNA molecule from other organism. These experiments have greatly increased public awareness of the potential for both useful and harmful manipulation of genes. In 1975, scientists who were working in this field stopped to consider the possible consequences of what they were doing. As a result of their deliberations, controls were on the types of experiment. Corporations now actively use transformed biological system to synthesize commercially useful products such as human insulin for diabetes, growth hormones and various immunological factors that may be useful in boosting resistance to disease including cancer. Several proteins including enzymes may also be synthesized for recombinant DNA technology. Recombinant DNA technology utilizes a number of enzymes, particularly restriction endonuclease from bacteria and DNA lysase from bacteria or bacteriophages, to produce extra genes into the cells with the help of vehicles known as vectors. Plasmids are a group of vectors which includes small circular cytoplasmic molecules of DNA and act as a extra chromosomal genes in bacteria. In case they attach them to chromosomes, they are known as episomes. Plasmids, when present, impart additionally properties to organize, for instance, resistance to certain antibiotics. Plasmids can be altered, for instance, plasmid PBR322 can be cleaved at a single specific site by ECO RI restrictions enzymes, which make staggered cut in a double helix leaving complementary single strand ends. If DNA fragment to be inserted is removed from its location on large DNA molecules by the means of some enzymes, it will have single stranded ends complementary to those of cut plasmids, and these two can be joined together by means of enzymes. DNA lysase as shown in figure 9.2. The new circular plasmids so formed can be taken up to small degree by bacteria E. coli because plasmids contains genes for resistance to antibiotics, ampicillin and tetracyclines. The bacteria containing the plasmids can be easily selected as shown in the following figure 9.2. Production of recombination of DNA molecules. Means these gene fragments A and plasmids in presence of DNA lysase, it give rise to according to the following reactions. In a similar synthetic, prokaryolytic and eukaryolytic genes can be incorporated into E. coli and other bacteria. As the bacteria is grown in culture, the inserted genes is replicated along with vector. The production of initial copies is called cloning. So this type we can say these long genes may be combined by the combined use of plasmids and K phages. This technique is known as cosmid cloning. In these techniques involving synthetic lambda DNA, multiple copies are linked via cos sites located at both the ends of chain to form a single long chain. COS sites undergo cleavage at 35 to 45 kb apart by the reaction of lambda packing enzymes, as a result of which lambda DNA gets packed into the phage heads. Now we can discuss the applications of recombinant DNA technique. A first application is in investigation of structure of genes. The recombinant DNA technology can be utilized in the determination of the structure of gene. For this purpose, if there is required of insertion and cloning of entire eukaryotic genes, then neither plasmid nor lambda phase or K phase vectors can be coped with the size of the inert. Size of eukaryotic genes is several hundred kb in length. However, yeast artificial chromosomes YS linear DNA segment derived from plasmids into which all basic function units of yeast chromosomes have been incorporated are the solutions to this problem. Now, second application is in the investigation of genome. Genome, that is entire genetic material of the cell, can be investigated by the application of recombinant DNA technology. Treatment of genome with restriction enzymes may cause cleavage at specific sites and may produce DNA segment which can be separated by electrophoresis. In agarose gels, depending upon their size, DNA material differ from person to person in human beings in respect of sequence of average on the base pair every 200 to 500. Difference in the sequence at a point may add or delete a cleavage site of certain restriction enzymes. This result of DNA segment. This process is known as restriction fragment length polymorphism, means RFLP, that it is shown in the figure 9.3. Third use is in forensic science. RELP analysis is used in forensic laboratories on human chromosomes on different sites. They are repeated non-coding sequences. Number of these tandem repeats vary from person to person. The length of restriction fragment from such a region of chromosomes also vary from person to person, except the cases of identical twins. Therefore, variation in number of random respects VNTR may be used in the identification of person. This technique produces genetic fingerprints. Specific probes for particular loci has been developed by the scientist Jeffrey and his co-workers, which provides two bands, one from chromosome derived from a mother and other from chromosome from father. Hair root, bloods, or semen found in clinical sport can be used in detection of criminals. As an evolutionary evidence, information about evolutionary relationship can be derived by application restriction enzymes technique of DNA from different species and races on living as well as dead ones. DNA polymorphism has been observed in beta globin gene cluster of individuals from 8 different human populations, visualizing Indian, Italian, North European, Thailand, African. So, next application is in the synthesis of proteins. Any vector may be used to insert new DNA into bacteria. So, next application in diagnosis of inherited disease. Chang and Can revealed that treatment with restriction enzymes second produce 0.2 and 1.15 kb fragment from the normal beta globin genes. So, this type of fragment of DNA through a process is known as northern blotting. Then, next application is in gene tracking and gene detection. Most of the equilibrium genomes contains non-coding sequence. These regions can undergo multiplications more easily than exon region RELP, resulting from mutation in non-coding regions are comparatively common. Next, in site direction mutagenesis, recombinant DNA technology can be used in site directed mutagenesis. For instance, relevant gene may be used in phase M13 and single standard circular DNA is isolated. Now we can discuss the site directed mutagenesis is also essential part of protein engineering of, for example, stability, regulation, specificity, and the properties of coenzymes. Trypsins, carboxypeptidase, and alcohols dehydrogenase as well as lactase dehydrogenase can be modified in this way. For instance, lactate dehydrogenase may be converted to malate dehydrogenase by repelling threonine 246 by glycerin and glutamitate 102 by arginine, but substitution of arginine 173 by glutamine in the same enzyme causes removal of the need of activation by fructose 1,6 diphosphate. And the last application is in the determination of primary structure of protein. Primary structure of protein can be determined directly by technique of DNA sequencing. For instance, DNA molecule can be synthesized from mRNA. Base sequence of this is related directly by means of genetic code to amino acids sequence of proteins. To remove any doubt about the identity of protein, cDNA may be used as probe to bind the corresponding mRNA. After purification, this is used to specify protein synthesis and to characterize it. So, thank you all of you. आज जो हमने ये एंजाइम्स की एप्लीकेशंस थी उसी को कंटिन्यू रखा है, और लगभग ये बायोऑर्गेनिक केमिस्ट्री की जो 2 यूनिट हैं ये आज हमने इस वीडियो लेक्चर के द्वारा जो है ख़त्म कर दिया। नेक्स्ट वीडियो लेक्चर के अंदर हम जो है ये बायोफिजिकल केमिस्ट्री स्टार्ट करेंगे। थैंक यू।